I would like to tell you about the vaccination failure because we are not so advanced like Denmark in the eradication of BVD. And this is the problem that we observed in some of the herds in Poland. So as you all know, vaccination is one of the key elements, and recently it is being indicated that without vaccination, you are not able to totally eradicate the problem from your herd. So after removing PI animals, you should start vaccination as a control measure and a prophylactic approach to protect your farm from the virus to enter the herd. And the aim of this study was to compare the serological response in the cattle herds where we observed the problems. Okay, so we observed some of the herds where there were PIs animals, and some of them removed the PIs, some of them did not, but they introduced the whole herd vaccination, and they observed the problem that the new PI cows were born despite vaccination. So we wanted to check the serological response and to identify what subtypes of BVD were present in those herds. So we had 5 herds, and 3 of them were the ones where the PIs were removed before the vaccination. So this was the correct approach. But there were 2 additional herds where PIs were not removed; they remained because the farmers thought that through the vaccination they would get rid of the viral shedders, and they didn't pay attention to removal of PIs. So they were vaccinating. They did not observe any clinical signs, but there were still PIs present in the herd. In all those herds, the calves and the heifers were kept together, so they had contact with each other. And in all herds, we used the same vaccine. Killed vaccine was used. Unfortunately, it was based on type BVD1A, which was never identified in Poland, so it was not matching the subtypes that we identified in Poland so far. So as you can see, there are 5 herds, and the herd sizes are indicated in the table, and the number of tested animals is more or less the same. The only thing about the age of animals is that in one herd, which is indicated as K, we had both heifers and adult animals, so 4 months old and 12 months old. So we could assess the cholesterol antibody response. The duration of vaccination was also various from 1 year to 6 years. And as I said, in 2 herds, OS1 and OS2, the PIs were not removed before vaccination, while in other herds, all animals, all PIs were removed. And there was only 1 herd indicated as K where clinical problems of respiratory system were observed. We use different techniques, like you can see, ELISA antibody, virus neutralization test, antigen detection by ELISA, and virus detection by PCR, and also sequencing and phylogenetic analysis of the isolates from the herds where the PIs were identified. So going to results, these are the results for the herds where BVD1B was identified. So on the left-hand side, you can see the box and whiskers plots where you see the value of 25 percentile, which is lower half of the positive results, and then 75%. This line here in the inside is the median value. Outliers are indicated as circles. So this is for 1A, which means the vaccine strain, and 1B is the isolate from the herd. And here on the right-hand side, you can see the presence of antibodies based on the virus neutralization titers from low values, so negative values below 10, titer between 10 and 20, 40, 80, and 160, 640. So the darkest ones are the ones which had the highest VN titers. So here is the herd A, where the vaccination was going only for 1 year. And as you can see, there was much higher prevalence of high antibodies for the field strain compared to vaccine strain. This herd is the one where the PIs were not removed. So you can see more or less similar picture for both vaccinal strain and for the field strain. And the same for another herd. These were actually two herds belonging to the same farm, but they were located in different places. So they were conducting this vaccination program for 6 years. And after the 6 years, they decided to identify PIs, and they identified from 5 to 7 PI animals in those herds, despite vaccination for so many years. This is herd K, where we also tested young animals, so 4 months old heifers. So as you can see, the presence of antibody titers of high value above 160 was much lower compared to animals 12 months old, which were already vaccinated. And we additionally tested here also presence of antibodies to other subtypes which were not present in the herd, because here in this herd, 1D was identified, subtype 1D, and here also you have comparable values for high titers for both vaccine and field strain, but surprisingly also substantial values for 1B, which was not present, and 2A, which was identified only in one herd in Poland so far. And another herd where 1B was identified, here you can see also more or less similar picture for both vaccine and field strain, and 2A was also tested with much lower values. So we try to identify the subtypes. You already seen it, so I just give you the bigger picture of the upper part of this phylogenetic tree, which was based on 5' UTR region. So here you have this vaccine that was used, Bovilis, which is 1A type, and here are the isolates indicated with the black dots, and the triangles indicate the vaccines available on Polish market. So it is much closer, this 1B type to Bovilis than 1D, which is in the lower part of the phylogenetic tree. Similar situation is observed also for NPRO region, where you can see that those 1D subtypes are much further from the vaccinal strain. So we also try to analyze the comparative strength of neutralization, which was established by Silveira. But as you can see, more than 40% of the samples didn't have a predominant titer for either vaccinal strain or the field strain, and only 16% of positive samples were predominated was 1A, and for 32% the titer again was the one that was detected in PI animals. Surprisingly, 10% of the remaining sera showed predominant titer for a subtype which was different from both vaccinal and PI strain. And within this category, 12 samples, which comprised around 17% of the all samples tested, had predominant titers for DVD2A subtype, which, as I mentioned earlier, was identified only in one herd in Poland in 2014. So the prevalence of PI animals was the highest in the herd where the 6-year vaccination was going on, but this was the herd where they were not removed before the vaccination. And the percentage of titers for heterologous strains was much lower than for homologous strains. And in 4-month-old calves, where the colostral immunity was still present, the values for samples was very low, with 50 and 60% of negative samples. And when we compare the sequence identity within 5' UTR region of vaccine and field strain, the subtype 1D, as I showed you on the phylogenetic tree, was located furthest from the sequences of the vaccine that was used in the herd tested. Thank you.